So I'm back. This is a long chapter. We're looking at a tour of the cell. In this video, we'll look at the structure and function of the Golgi and lysosomes. So let's scroll down to the right slide. Okay, the Golgi apparatus is, sometimes it's just called the Golgi or the Golgi apparatus, is a part of the endomembrane system. So we need to write that in, I think. It's a part of the endomembrane system, the system of internal membranes that help to make molecules and move them to their right location, right? And the Golgi apparatus is one of the coolest looking structures in the cell. There can be one of these in a cell or hundreds in a cell, but basically they're a network of flattened disc-shaped structures that are unconnected to each other. So each of these has a membrane around it. Each of these little sacs has a membrane around it and then has a space inside. That space inside is called the lumen, or sometimes it's called the cisternae. And sometimes you'll see little tiny vesicles kind of off to the side or budding off of these unconnected membranes. Okay, so it might have 5 or 6 flattened sacs. And the question is, what's happening here? Well, first thing to note is there are kind of sidedness or polarity to the Golgi apparatus. The side that receives vesicles coming from the rough endoplasmic reticulum is called the cis side of the Golgi apparatus. And materials tend to move from the cis side, so these little modified proteins that aren't quite fully mature proteins yet, arriving from, in little tiny vesicles, arriving from the ER, these molecules are going to move through the Golgi apparatus and eventually leave the Golgi apparatus at the trans side of the apparatus, right? That's the shipping side, if you will, of the apparatus. People call the Golgi the post office of the cell, right? So let's take a look at how molecules move through the Golgi and then what the Golgi is doing to these molecules. Now, let's redraw the Golgi apparatus, this series of flattened discs or sacs, right, that are unconnected to each other. One side is the cis side, where vesicles are coming in. They are coming from the ER, so maybe you should put that they're coming from the rough endoplasmic reticulum here. And when they arrive at the cis side, these vesicles release their contents into the lumen of the first sac on the cis side of the Golgi apparatus. Each sac has sets of enzymes that can change or modify or identify the proteins that are coming in. And as these proteins that are coming in get modified, they move, and we're not really sure how. They might kind of bud off in little vesicles to the next sac, and then bud off again to the next sac, and eventually arrive in the last sac of the Golgi apparatus. Some people think that each sac kind of changes and becomes the next sac. We're not exactly sure, but we do know that molecules are moving or transversing, crossing through this Golgi apparatus from the cis side to the trans side. Okay? So there's this functional movement of molecules. What's happening to these molecules as they move through the Golgi? I have 3 basic functions that we want to know. One of them is molecular modification. So these proteins aren't quite ready to do their job yet. So they might need to have a sugar added to them or a small sugar taken away. So there's modification of the proteins, and what comes out of this is essentially a protein that has some sugar attached, a glycoprotein. Second, these glycoproteins are identified They are given ID tags, molecular tags, like a phosphate group is added or some other tag is added to them that's going to help identify where they're going. These molecules then leave the transface of our Golgi apparatus, and they tend to go in one of three places. They might be headed in little vesicles toward the membrane to become membrane proteins. They might be headed in a little vesicle to be secreted. Maybe they're a hormone, like insulin, that needs to be shipped out of the membrane. Or they might end up in what's called a lysosome. And that lysosome will operate inside the cell. So the Golgi tends to complete the maturation of the glycoproteins and then identify or tag them so that they can be shipped to their appropriate location. Another thing that happens in the Golgi that's unrelated to these glycoproteins is the synthesis of polysaccharides or carbohydrates, like cellulose is made in Golgi. So Golgi have a pretty complex set of jobs to do. Okay, next up comes the lysosome. So the lysosome, we think, buds off the Golgi apparatus, kind of forms a little tiny vesicle. And let's take a look at what the lysosome does, thinking about the structure and functions again. Right? A lysosome is a very small vesicle. That's why we call it a vesicle. That's maybe 1 micrometer in size. And it's a little round sac, basically, that is filled with digestive enzymes. We call these enzymes hydrolytic because they use water to break, or to hydrolytic. Oops, there we go. Okay? Hydrolytic enzymes. There are in humans something like 40 different types of hydrolytic enzymes inside our lysosomes in animals. And the hydrolytic enzymes do the job of digesting big molecules and breaking them into smaller molecules. So taking proteins and breaking them into amino acids, or taking complex carbohydrates and breaking them into their simple monomers like glucose, or taking fats and breaking them into their components. And there are different kinds of enzymes. One other thing about these lysosomes in terms of their structure is they're very acidic. The lysosome has a pH of around 5. The bigger cell has a pH of around 7. It becomes acidic because the lysosome actually pumps protons into the cell, increasing the concentration of protons inside the lysosome, making it very acidic. And it turns out these hydrolytic enzymes only function at about a pH of 5. They don't function anywhere else. And that's a good thing, because if these hydrolytic enzymes left the lysosome, they could start destroying the cytosol, destroying things in the cytosol. But instead, they only work at a pH of 5, and the lysosome sits at a pH of 5. I like this. Some of the functions of a lysosome, well, you can probably imagine, they do the job of digesting things. So sometimes in some organisms, an organism will take in food in a vacuole, and the lysosome will sidle up to that food vacuole and release its enzymes and digest the food, making it a source of nutrition for the animal, for the cell. Sometimes the lysosome helps to break down old, used parts inside the cell. Turns out that our cells have parts that get old, like a mitochondrion that gets old, or a chloroplast that gets old, and the lysosome will sidle up to it, release its enzymes, and digest it, and the cell can recycle those parts. In fact, sometimes I think of the lysosome as being a recycling center, and pretty important for a cell. Okay, I have a note here about Tay-Sachs disease and how it's related to lysosome function. Here's just another picture of how lysosomes can help destroy viruses and bacteria. in a type of cell we call macrophages. Macrophages are part of our immune system and clearly very important to us. But what they do is they find and gobble up, essentially, viruses or bacteria. How do they do that? Well, they're filled with lysosomes. And so they'll take in a virus, and the lysosomes will digest that virus, and the cells then get rid of all the junk that they don't want. So lysosomes can be really important. Okay, here's a response question that you should think about. What do you think would happen if one of the enzymes that normally functions in a lysosome doesn't work? In other words, you have many different types of enzymes in your lysosomes, but what if one of them is broken? What would happen to the cell? People with Tay-Sachs disease have an enzyme that's broken, and that enzyme normally exists inside lysosomes. People with Tay-Sachs disease don't usually live to be more than 4 or 5 years old. It's a horrible genetic disease that when someone inherits 2 mutant copies of a gene, they can't make the enzyme beta-hexohexosideriminidase hexa, and that gene codes for an enzyme. The gene codes for an enzyme that functions inside lysosomes. You can check out, if you want to read up on Tay-Sachs disease, you can probably address this question. This question will show up on that extra credit assignment I was talking about. So check it out. And that's lysosomes and the Golgi apparatus.